So it's my great pleasure to introduce Ivana Bozic. She's retaliating for the visit I made to her department a few months ago, which was great fun. So I hope we'll treat you as well as you treated me. So Ivana is an associate professor in the Department of Applied Math at the University of Washington. And she's at the moment, I think, on sabbatical in the Hutch, the Fred Hutchinson Cancer Center. So she got her BSc and MA in mathematics from the University of Belgrade, and her PhD in math from Harvard. And she's won a number of prizes, the most recent of which is the 2023 Akira Okubo Prize from the Society for Mathematical Biology. So she focuses on using mathematics to understand the evolution of cancers. And today she's going to tell us how she does it.
Thank you so much. Thank you so much, Simon, for the invitation. Thank you for visiting us in Seattle a few months ago. And it's actually my first time at Columbia and at IICD, so it's great to be here. And it's great that such an institute exists somewhere in the world. We've been talking how people who are interested in cancer from a multidisciplinary point of view are always at very different places, even at the same university. As you saw in my notes, my research is focused on quantifying and understanding cancer evolution as it happens in patients. And by that, I mean, I would like to understand what are the relevant models for how cancer is initiated, how it progresses, how it responds to therapy, what are the key relevant parameters for cancer in humans and specific cancer type in humans, what are the timescales over which this process is happening, and also if we are talking about mutations that are occurring on the way to cancer, why do they seem to happen in a particular order in many cancer types and not just in any way possible, or it seems that is what is going on. What complicates our efforts to understand cancer evolution, accumulation of driver mutations in different cancer types, and even within a single cancer type? Well, one of the many things is the huge heterogeneity we see wherever we look in cancer. If we look into driver mutations, even in a single cancer type, we'll see that there are many, a few that are mutated at a significant frequency, but many that are mutated at lower frequency. And as you go further down the frequency, it gets very difficult to distinguish by statistics alone mutations that are drivers versus those that don't have an effect on the phenotype. Here you can see some of the most common cancer drivers in general. But as I said, situation is complicated even when focusing on a single cancer type. And what makes it even more complicated is that once we actually sequence cancer genomes and look what are the mutations that are found there that were not present in the germline, well, most of these mutations are not even drivers, they are passenger mutations. So if we want to understand the evolution of the cancer genome, we should also, and understand sequencing data, we should also try to take into account passenger mutations as well. But all is not lost. So let's see what are some of the steps we can take if we want to understand how driver mutations, to start with, accumulate in cancer. And if we want to make models that are specific to the underlying biology, we should take into account that some of the cancer driver genes are tumor suppressors versus others, where others are oncogenes. And they have very different functions in the body, and they are also mutated to become cancer drivers in a different way. So this is something that we want to take into account when forming our mathematical models. And so oncogenes or proto-oncogenes, typically their function, healthy function, has nothing to do with cancer. But if they get a mutation at a specific position or a specific few hotspot position in that gene, they become activated and act as oncogenes and drive human cancer. So a single mutation at a small number of positions is sufficient. Whereas tumor suppressor genes, their normal function is to prevent us from getting cancer. So they need to be fully inactivated, typically to act as cancer drivers. And that means that both copies of that gene need to be inactivated, either by mutation or by something we call loss of heterozygosity, where basically one copy of that gene is lost. And so for that, we need kind of two events for inactivation to happen. Then the next question is, how many driver mutations do we need for cancer? How many mutations are present in a typical cancer? And this graph is trying to give some insight into that. And here it's we see several cancer types and the number of oncogene mutations only in red, and also all drivers, which is oncogene plus tumor suppressor genes. And I'm mostly interested today in colorectal cancer. So what we see a distribution of the number of known drivers present in their genomes, but typically we see a single oncogene and a total of 3 driver mutations. So roughly 1 oncogene and 2 tumor suppressor genes. But of course, understanding this variation fully would be great, but we are not there yet. And then this paper and some other works seem to point to the fact that 3 driver mutations may be sufficient for the appearance of, in particular, colorectal cancer. And this is, again, 1 oncogene and 2 tumor suppressor genes. And of course, we do not know if this would be true in all colorectal cancers, but this will be the assumption that I will be using going forward. Ideally, we would love to know more precisely the numbers. But this idea of 3 driver genes for colorectal cancer also agrees pretty well with the famous Vogelgram, which talks about the stages of development from healthy colonic tissue to colorectal malignancy as the cells are gaining more and more driver mutations. And so here on the left, we see the normal architecture of the human colon, which is by this model supposed to be followed by inactivation of APC as the first change, as the first genetic change, which leads to a formation of an early adenoma. This is not malignant, this is a benign tumor, but it can get more aggressive by getting another mutation, such as KRAS, forming a large adenoma. And then this model supposes that the last step on the way to colorectal cancer is another driver mutation, which could be something like a mutation in the tumor suppressor TP53. So we don't know that this is the order of mutation for certain, because we cannot observe cancer evolution in patients, but this is inferred from the fact that if you find small adenomas and sequence them, typically you will only find APC mutations. It still doesn't mean that there cannot be some occult small lesions harboring different mutations that maybe once they get something else would lead to cancer. But this is kind of what we think is the order of mutations on the way to colorectal cancer, roughly. So we wanted to try to understand and develop a mathematical model that would take into account some of this knowledge about what causes colorectal cancer. And we were lucky that when we did this study a few years ago, a lot of the parameters that we needed for our model have been experimentally determined in humans. And this is a collaboration with my former postdoc, Shay Patterson, who is now at the University of Manchester, working with David Wedge and Hans Clevers. And so I will be talking for the next few or more minutes about this paper, in which we wanted to focus on the 3 most common cancer drivers in colorectal cancer, which are these 3. APC, TP53, which are tumor suppressors and need 2 steps to be inactivated, and KRAS, which is an oncogene, so needs a single mutation so to be activated. As I said in the beginning, there's huge heterogeneity of driver mutations within different patients. So these three mutations are present together in about 15% of colorectal cancer patients. So this is what we are focusing on, trying to explain this fraction of colorectal cancers. And by taking into account that we have 2 tumor suppressors and 1 oncogene, we actually have a 5-step process on the way from a healthy cell, which is here on the left, to a fully malignant cell, which is here on top right. And we get there by any 5-step path along this cube of genotypes. So basically, we just need 2 tumor suppressors inactivated in any way, and then in any order, and then an oncogene as well. And even though there is an order that is kind of what we think the order of mutation is in colorectal cancer, we were really interested in understanding, is there something intrinsic about that order, or what would happen if we assume any other order is possible? What would that lead us to? Would we get the same or something different? And so we actually have, I think, 50 genotypes here. It's a little even more complex than this because there's multiple ways to inactivate a tumor suppressor gene. And we have like hundreds of paths from healthy to malignancy. And please stop me at any point for questions. So we have our network of genotypes, or graph of genotypes if you want. But what happens? We also have cells, or in our case, we actually have colorectal crypts as units of selection. Because the colon consists of about 10 to the 8 healthy crypts, each of them having about 2,000 cells in total. But what happens in a crypt is mostly determined by a small number of stem cells that reside at the bottom of that crypt. And the simplifying assumption we will make here is that once the mutation appears in a stem cell in a crypt, it will either be washed out or lost, or it will take over the crypt within a reasonable amount of time, so that we can kind of neglect what happens inside the crypt and just focus on crypts as our units of selection. So we will start with our 10 to the 8 healthy crypts. So what will happen to them as they are getting these mutations? Well, as I said, luckily in the last, well, almost a decade now, people have actually measured what happens in human colon once APC is fully inactivated. And they actually found that it leads an increase in the crypt fission, or basically a division rate of crypts, which is more or less 0 in healthy tissue, or very close to it, to rates of 0.2 per year. People have also more recently measured what happens once a KRAS is activated in human colon, and that leads to, again, an increase in crypt fission, but a smaller one. And then TP53, although I think this wasn't in humans, it was in mice, but it was shown not to lead to selective advantage of it on its own as the first mutation in colorectal cancer. So we are gonna take that assumption in our modeling.
Oh, I had a question about the 2 hits you need for the tumor suppressors. Is there like an effect where if you have just one, there's a slight selective advantage, or do you like definitely need both to get the advantage?
So in a sense, both. So there's a subtle effect that we take into account. But so there was a paper, I don't know, 10 years ago, that looked into, again, mice crypts and showed that inactivating a single copy of APC leads to higher chance of that mutation taking over the entire crypt. So we take that into account when we calculate our mutation rates. But For us now, a crypt that has just a single copy inactivated does not have an advantage. So that kind of deals with our that deals with our growth rates in the model. And what about mutation rates? What are the rates of transition from one genotype to another? And again, we were lucky that recently, or as years are going by, this is less and less recent, but Hans Claris's group was able to calculate the rate at which mutations accumulate in the genome of colorectal stem cells in healthy colon. And that rate is about 40 mutations per genome per year. So we use that as our baseline point mutation rate. And then we also calculate the number of specific driver positions in our 3 genes of question to get mutation rates. And so here I have slightly different genes, but the idea is that genes like KRAS, which are, or PIK3CA, which are oncogenes, have only a small specific number of positions where they can be mutated. So we count that for KRAS, whereas genes like TP53 or APC are tumor suppressors. So we just need a mutation that will basically make a stop codon and inactivate that copy. And so we calculate the number of positions that, if mutated, would lead to such a change. So I think this is when it comes to our parameters, but for this project, we really wanted to parameterize this with relevant human parameter values. We study the model in 2 ways. The first way is through computer simulations. We start at age 0 with 10 to the 8 healthy crypts, so we can neglect the development. And we track the numbers of crypts with all 50 genotypes, as well as paths that are taken by individual crypts. And there are 270 possible paths. We use both Gillespie and tau leaping algorithms, Gillespie, which is exact, and tau leaping, which is faster, but still very similar. We also try to get some analytic insight into the process by using what we call a mean field approximation, but it's kind of a semi-deterministic approximation where we calculate expected values of cells in each pre-malignant genotype, and then use that to find the time to malignancy. And to kind of complicate things a little more, we take into account 3 scenarios. The first scenario is where we assume that all drivers are neutral. There's a lot of talk about neutral evolution, whether that can happen prior to malignancy, or is this something that happens after malignancy only? So we wanted to see, can, just by collecting neutral drivers that are neutral pre-malignancy, can one get something that is reasonable? Then what if only APC has a division advantage in colorectal crypts? And then the full model that kind of corresponds to what is found experimentally, where both APC and KRAS have a division advantage. And these are relatively simple estimate approximations for probability of malignancy by time t. Okay, so now I'm plotting here the 3 lines that correspond to these 3 formulas that I showed on the previous slides. The blue line is probability of malignancy as a function of age in the fully neutral model. And here we see that at age 80, in the fully neutral model, probability of colorectal malignancy is on the order of 10 to the negative 9. Or, which is, of course, much less than we know what the incidence or prevalence of colorectal cancer is in humans. This is a line where APC only has advantage. And again, this is on the order of 10 to the negative 5, still lower than the real incidence. And actually, the real probability of having colorectal cancer by age 80 or by developing it is shown here in this single data point that is a kind of an epidemiological data point on prevalence of colorectal cancer, and that's on, I think, 4% total, but we're only focusing on 15% of those, so less than 1%. And we see that this is in agreement mostly with our green line, which is assuming both APC and KRAS have advantage and TP53 does not. And then if you're interested in how the simulations and the approximations agree, well, they agree well, unless you start having larger growth advantages like here, and then they start to deviate at later ages. But our simulations are in good agreement with this kind of lifetime prevalence of colorectal cancer. So our idea was not to fit the entire curve of cancer incidence. The model is not, I think, sophisticated enough to capture everything there, but to show that we are, by taking into account biological knowledge, measured parameters, that we can get something quite reasonable in terms of qualitatively and quantitatively in terms of colorectal cancer prevalence. And because we saw this discrepancy in our computer simulations that are exact, and in the our first approach at calculating this analytically, we wanted to explore this in more detail. And in this recent paper done by graduate students Ruibo and Obina, we were able to get very good formulas that are very accurate for probability of malignancy. However, they are much more complicated, so I'm not showing them here. I'm just showing how well they fit with the simulations. So one of the key things we wanted to see is if by taking biological knowledge plus measured parameters, we can take out something that resembles colorectal cancer incidence. Another question we wanted to ask was the order of driver mutations. And now we said, if we focus on all paths that reach malignancy by age 80, what is the most likely order of driver mutations along those paths? So not all paths in general, we specify that they have to reach malignancy by age 80. And if we look into it that way, we see that in about 2/3 of cases, APC was the first mutation to be collected, followed by KRAS, followed by TP53, which is interesting and kind of exciting because it is basically what we think is happening in patients as well. Although we do see in about 1/3 of the patients that KRAS could be the first one, followed by APC. And the reason is that KRAS needs only a single hit to become a driver. So in our model, there are numerous very small lesions that have KRAS. And each of them has a very small chance to progress to cancer, but there's so many of them, so there's some non-negligible chance that one of them will actually progress. Yes?
How do you determine the order of the mutations?
Yes, so we look at, we run our simulations many times, and then only pick paths that reach malignancy by age 80. And so they all look like this. They're all 5-step paths where you collected 5 steps by age 80, and then we'll look at the order. But we were interested, why do we have this order? And again, we were able to somewhat answer this question in this semi-deterministic setting. So kind of an analytic approximation of the process, and the equations are not that important. But what is important is that in our model, the product of mutation rates is constant. Because even though you can get to the same way in different paths, kind of mutation rates along the path, if you multiply them, they are constant. And so we can show that the probability that a certain path is taken is maximized when basically you maximize fitness as you go along the path. So you first collect the most potent driver, then the second potent, and so on. So kind of in our model, it's so kind of what we see in patients can be explained not by there being something specific to APC that it has to be first, but that potentially just we often see it first because it is the most potent. If something collects a different driver first, it will not reach malignancy in time.
So I think that there is some work on evolution in bacteria where you have origin strain and target strain, and people looked at the different paths that evolution is taking in that context. Is that I don't know.
I don't know that work.
Okay.
And would love to know more. So yeah, if you can think of a paper to send.
That will require some archaeological digging, but yeah.
But that sounds exciting. And my student would be super interested, so. And another thing that was very curious here is that once we did this analytic analysis, we also noticed that in this setting, the order of driver mutations is independent of mutation rates, which was curious. I expected that fitness advantages would play the largest part, but the mutation rates would still be important. However, here we can see that because their product is the same, they kind of all cancel out. And the only thing that matters is the fitness, which is also very interesting, at least in my opinion. So we saw that in analytic calculations, but we also saw it in simulations, where here we show relative probability of different paths with our original rates. And then we multiply our rates by 2, divide all of them by 2. And when I say rates, these are mutation rates. And then here we only multiply one of the rates by 10 to have it non-symmetric. And we still see the very similar up to stochastic fluctuations, probability of paths in all these settings. Any questions before?
Yeah, wait, sorry, can you explain again, what are the different mutation rates? When you say the different, you explained you more than one of them, what are the different ones again?
In the model?
Yeah.
Yes, so we have basically as you go. Yeah, so to go from any node on this network to any neighboring node, there's a mutation rate or a transition rate.
Gotcha.
Yeah.
And that transition rate was determined by like counting the number of potential mutations.
Correct.
Okay.
Correct. But we show that the order is unaffected even if we say multiply them all by some factor or divide them. Okay. So for the second half of my talk, I will be talking about a different cancer type, which is chronic lymphocytic leukemia. And most of this work was done in collaboration with the labs of Kathy Wu from the Dana-Farber Cancer Institute and Gary Gatz from the Broad Institute in Boston. So first, a bit of intro into CLL. It's a B-cell malignancy, and a tumor burden can be tracked very well in this cancer by just taking a simple blood count and looking at the number of white blood cells. So this is a typical progression of CLL over years. it is described by clinical heterogeneity. It can be very aggressive in some patients, but it can be also very indolent in a lot of patients. So initially, what is typically done is called watch and wait. So even though CLL is diagnosed, it would not be treated unless the patients develop symptoms or it starts growing too fast and so on. So what is very interesting about CLL as a case study for mathematical modeling is that we can observe it unperturbed by therapy over many years in patients, which is very unlikely any other cancer that I know of. And so you can see over the years how the white blood cell count starts rising. When it becomes too much, treatment is initiated. Typically, this is combination chemoimmunotherapy, which is called FCR. And it's very effective, so it would lead to good response in many patients, but a lot of them will develop resistance. And then there's a lot of new drugs, like targeted therapies targeting different signaling pathways that are then these patients are taking. So we want to understand, I want to kind of understand all parts of this process, but I will mostly be focusing on this one, understanding natural progression of CLL. And so we were very lucky that our collaborators had this data set of CLL patients that were followed before their first treatment, and we had longitudinal sequencing samples, and we wanted to understand their genetics. However, even before we looked into that, we looked at the white blood cell count as a measure of tumor burden and saw something very interesting for me as a applied mathematician or a mathematical biologist, and it was that in many patients, the white blood cell counts followed these two very well-known simple growth curves, namely exponential and logistic growth. And it's super exciting looking at patient data to see anything that makes sense. So this was very exciting, and we wanted to see, well, is this really happening in patients, and does it correlate with anything that we care about? So let me first explain our data. We had 21 patients in our discovery cohort. They were all treated at some point with this chemoimmunotherapy. But some of them were followed for like 18 years from diagnosis to treatment, whereas for some it only took 2 years from diagnosis to treatment. And these filled dots you see here are points where we had sequencing available. And then we kind of wanted to fit their white blood cell counts to one of our models, and we kind of made a full Bayesian model that does that based on the posterior of the carrying capacity. But we did see that most of our patients fall very nicely into the both either exponential or logistic growth category, whereas a few of them were in this indeterminate category, typically because our follow-up was not long enough, but some were also a little weird. So what was also very interesting is that if we looked at how long it took from diagnosis to treatment, the logistically growing CLLs tended to have longer time to treatment compared to exponentially growing CLLs, which had a shorter time. And this is something that we also looked into a larger cohort to confirm, and actually found basically the same thing. Here, it's a bit limited by the number of patients. But then we also look at the underlying genetics. And so here, we have our patient here in these different columns, logistic, indeterminate, and exponential. And for each patient, we have several of these columns because each column is basically a sequencing time point. And here we also show known cancer drivers present in these patients, as well as their clonal fraction in the tumor. So there are several interesting things that one can see here. One is that logistically growing CLLs tend to have on average a single driver mutation. Whereas exponentially growing CLLs tend to have many more, on average 4. And this is also something that we confirmed in a larger cohort. What is also very interesting to me is that even though some patients are followed for a decade or more, we don't see much clonal evolution. If we see a driver present at the first time point, it will typically be seen at the last, and vice versa. We don't really see new drivers popping out. So it seems that the stage is set prior to diagnosis for these patients, and it will play out later over many years. Because we had longitudinal samples, it was relatively easy to calculate growth rates of different subclones in these patients. And what we were also interested in, selective advantage provided by known drivers. And what we found is that strongest advantage was provided by second hits in tumor suppressor genes TP53 and ADM, which was also kind of interesting that and kind of shows that one needs to take into account if you're dealing with a tumor suppressor gene, kind of trying to see whether 1 or 2 copies of that gene are inactivated. And kind of one other thing that was interesting in this study is that we found association between the growth pattern and the evolution following or playing out following treatment. So this is again, a treatment with combination chemoimmunotherapy. And we saw that in exponentially growing patients, like patient 6, you can see here the subclonal structure of this patient and the treatment administered around here. You can see the white blood cell count going down. Then there's a relapse, but the relapse looks subclonally very different from the original tumor. On the other hand, in logistically growing patients like this one, what comes back after treatment looks identical genetically to the original tumor. And we had a limited data set of patients where we had both pre and post-treatment sequencing done, but we see that trend across all of them. So that begs the question of what, are exponentially growing CLLs just more flexible evolutionarily compared to more rigid logistically growing ones. And in this follow-up study with my former graduate student, Nathan Lee, who is now at Bifold in Berlin, we tried to look a little more closely into this data and try to infer more parameters than just the growth rates and the selective advantages. And we did that by studying a multi-type branching process that takes into account different subclonal populations that have driver mutations. Here we are only depicting a single subclone, but we generalize it to the case of many subclones. And also by taking into account the accumulation of passenger mutations. And in this model, we'll leverage the fact that we have sequencing data available at 2 pre-treatment time points, which we had in this case, and which is a little trickier in general. But in that case, one can actually get very simple analytic estimates for many parameters of the process, including mutation rates, growth rates, and timings. So, as I said, basically, the presence of passenger mutations complicates significantly the reconstruction of tumor evolution, but is actually helpful if you want to infer mutation rates as well as timings. And I will skip kind of the derivations and the formulas. You can look at the paper if you're interested in more. But what I think was the most interesting in this study is that we could calculate, and here you see this is the patient age. These three arrows are the times of sequencing, and the white dashed line is the diagnosis. And we estimate the time of the parental subclone of CLL in these patients. In basically all cases, that time is very early, within the first or second decade of life. And this is kind of in agreement with what is found in many different cancer types, that it seems that it could be decades from the first tumor cell to actually a diagnosed cancer. Of course, one caveat here is that we used fixed mutation rates as well as fixed growth rates. With that, that should be taken into account, but nevertheless, very interesting that the seed seems to be planted very early. Okay, and so kind of instead of a conclusion for my last several minutes, I want to talk about some older papers and connect them to something that is very relevant today. And instead of talking about cancer evolution prior to treatment, now I will focus more on what happens after therapy, and in particular, resistance to targeted therapy. So these are two papers from almost a decade ago now. I can't believe how much time has passed, where we looked at the models for evolution of resistance prior and following targeted treatments. These treatments are very amenable for mathematical modeling and analysis, because we know that typically resistance to targeted treatments happens due to alterations in either the target genes or some related genes. So it's very well understood. This is in contrast to, say, resistance to chemotherapy, which is much less well understood and often not genetic, or we don't know how it connects to genetics. So in this first paper, we showed that if you had, well, first that if you have a single targeted therapy that has any possible potential resistance mutations, it is likely to fail. It is doomed to fail, basically. And if you have, if you want to have any chance of success, you want to combine different targeted therapies that have completely opposite modes of resistance and no cross resistance. So here we have two therapies like that, where if we combine them, we get a cure in almost all cases. But if they were given sequentially, they will fail one by one, because each of them will be opposed by resistance. And then we went into a little more detail of understanding this pre-existing resistance in this other paper, where we basically showed that if you have a population of 1 billion or close to 1 billion cells, which you often do have in cancer, you are certain to have multiple resistance mutations present at the start of therapy. They will not be detectable typically. They will be present in maybe 1 in 1 million cells, but they will be there, and there will be multiple different mutations. And as the therapy is starting, they will rise in frequency because other cells will be killed by therapy. And we saw some confirmation of this in this paper from 2016, again in collaboration with Kathy Wu and Gary Gatz, where we followed a patient this is again a white blood cell count. This is chronic lymphocytic leukemia, treated with this chemoimmunotherapy, which then relapsed after 3 years, at which point it was given this targeted therapy that was new at that time, which was a BTK inhibitor, ibrutinib. But then after a few years, again, the patient developed relapse to this therapy. But what we see once we sequence at multiple time points is that the original population that was blue was completely replaced by these 4 different, and we saw that there are different resistance subclones that each have different point mutations in the same gene, PLCG2. And now we know that this gene, mutation in this gene, provide resistance to this targeted therapy. And we showed by mathematical calculations and droplet sequencing that these mutations were indeed pre-existing at the start of therapy, although at very low cell numbers, maybe in 100 or 1,000 cells or less. And so this brings me to a more recent study in collaboration with two colleagues from University of Washington, Rosanna Rizquez and Cecilia Young, where we looked at patients that have CLL as they progress through different treatments. So we are not seeing everything. Typically, they were all given this chemoimmunotherapy in the beginning. We see part of their treatment. This is venetoclax, another targeted therapy, then acalabrutinib, which is just also a BTK inhibitor similar to ibrutinib, but maybe a newer one, and so on. What is interesting here and kind of matches with our theoretical predictions from many years ago is that after treatment with BCL2 inhibitor venetoclax, we start seeing mutations at very low frequencies in that gene. And one thing I didn't say is that here we did duplex sequencing, which can look at very low frequencies. And then, but there were no mutations in BTK or PLCG2, which give resistance to BTK inhibitors. However, after treatment with BTK inhibitors, at this later time point, we see mutations multiple again in both of these genes. So basically, exactly as we would expect from our theoretical predictions. We see a similar or even more interesting thing in this other patient where we have combination treatments for a while of two inhibitors, a BTK inhibitor acalabrutinib and BCL2 inhibitor venetoclax. And after treatment with those two, we don't see any resistance mutations. So that's kind of anecdotal, but again, along the lines of what we would predict. And why this is not just anecdotal evidence, what I found and why I added this to the top was this very recent report from ASH this year, where actually a combination of these two targeted therapies, which target very different signaling pathways in CLL, was actually superior to this combination chemoimmunotherapy, which is typically the first, the standard of care. And what is even more interesting for kind of a mathematically oriented audience interested in cancer, in this study, they actually directed duration of treatment or personalized it for each patient based on whether the patient had minimum residual disease or not, and at which point it stopped having any evidence of disease. So I think this is super exciting, both because we have a successful combination of two very different combination therapies, and because of this personalized approach to duration of treatment, where I think mathematical modeling can be very helpful. And that's all I had for today. These are some of my collaborators, my funders. Thank you for having me.
So I think we have time for questions. Hang on, Carla. Is there anyone online with any questions?
Not so far.
Okay.
Thank you. It's a great talk, and I've read so many of your papers, so I have a lot of questions. For your colorectal model, if you assume that the p53 doesn't have any effect on fitness, why is it included in the model?
Yes, that's a very good point. p53 has a huge effect on fitness if it's collected in order to get to the malignant state. So once it's collected as a third driver, it will have huge effects on the fact that cells can tolerate more copy number changes, just everything can be much messier, but it doesn't make a big difference at the beginning. And so because we only cared about the point of malignancy, we didn't include the strong fitness effects that TP53 is expected to have in later stages.
I think one of your predictions from the model is that you would expect the first driver to be APEC, more likely than KRAS.
Yes.
So then from that, you would assume that in the cancer samples, you would see more cases with APEC and not KRAS than cases with KRAS and not APEC. Is that something that you have?
Yes. So yes, you can look early on at these benign adenomas, and you see more APC in them. So that's one line of evidence. Typically, APC and KRAS both will be clonal in the cancer. So they will be the truncal mutation. Although APC is present in around, mutated in around 80% of colorectal cancer, and KRAS in maybe 50%. So it is more often mutated. So it could be, it could be argued from that point of view, but typically they would both be clonal.
And finally, in the CLL, when you compare between the treatment between exponential and logistic cases, I assume the treatment is the same for the cases.
Yes, so that's a subtle point. Let me see if I need it's here. Yeah, so in the discovery cohort, all patients were treated with the same combination chemoimmunotherapy as first treatment. And here, there's a trend towards shorter time to treatment in exponential category. When we went to the discovery cohort, the larger cohort where we wanted to confirm our findings, that cohort contains CLL patients that were treated and those that never needed treatment. So kind of contained also patients that had more indolent disease. And there we actually looked for the need for treatment as a variable that correlates with the growth pattern. And we actually found that exponential patients will need treatment at a very high, with a very high probability, whereas logistically growing patients will not necessitate treatment with a very high probability.
I mean the results on slide 26. So I assume the treatment is the same.
Yes, yes.
So one possible explanation for why you would see more of the same clones in the logistic is, I assume chemotherapy targets more the cells that divide faster, right? So that in your exponential case, those cells, I assume, divide faster. So they would be killed more effectively.
Right. So if I understand correctly, you're thinking maybe all the subclones here have the same growth rate, so they are all killed at the same.
Well, the clones on the right would have a lower division rate than the clones on the left.
Right. Right. But one key issue is the differential killing. Here, both the green and the black and the orange clones are killed at the same rate. Whereas here, this blue clone is killed at a much higher rate compared to the orange clone.
Yes.
Considering exponential and logistic growth kinetics, you consider Gompertzian kinetics rather than logistic kinetics, which might lead to understanding of the cause of the limit to growth and to the genetic distribution.
That's also a good point. We didn't consider Gompertzian growth, even though it's very similar to logistic and possibly could fit these patients, the logistic patients as well. We really wanted to understand the difference between exponential and logistic, and in our framework, we basically consider a generalized logistic model with exponential as a special case of infinite carrying capacity. So it was easy to classify this within a like the same framework. But it's interesting to look into Gompertzian as well.
Well, excuse me, any more
I
questions?
have a technical question, maybe I missed it during your talk for the colorectal cancer. Everything that you showed was on simulated data. Is that correct?
So inputs for our model, the values of parameters come from experimental studies, but our what we did is purely computational and analytic.
Okay, so for example, the result that you came to ABC coming before KRAS, that's all from your simulation, from your
Correct.
computational work. And if you had to like try and confirm that in real data and in real life, do you have any idea how you would do that?
You cannot completely confirm it in humans because you cannot observe the process.
Yeah.
But I think the consensus in the field is that that is the order in which things happen. And I already mentioned some of the things why that is.
Okay, thank you.
Okay, thank you very much, Ivana, for a fantastic talk. That was great.
Thank you.
Thanks.